Okay, introducing KJ to today's tails topic, which is can I spray during flowering? All right, over to you, KJ. You've got the floor. Let's dazzle us with your brilliance.
Awesome, guys. Good morning. Let's talk about can I spray during flowering? I'm a bit sad that there's few people on the panel today, but we'll just make the best out of the situation. Topics to cover today. We're going to talk about reproductive stages that include flowering, pollination, cell division. It's a bit of a crossover from what Emily has presented last week, but I think it's very important to address or understand those stages a little bit better. And again, key nutrients during those reproductive stages, soil applied nutrients, why we need to supply nutrients via foliar, factors that can impact the uptake of the foliar nutrients, common concerns that growers come across during spraying during the flowering, and then some key take-home messages from the presentation. So obviously some of us would have been familiar with the slide from the YouTube video that Smithy and Joe put in together. So I've just taken a bit of information from that. So stages of plant nutrient that are important in the physiological stages, we are aware of all of these ones, but we're going to mainly focus on the bloom, pollination, and cell division stages. It's very critical to understand the nutrient requirement of the plants during these stages for this presentation. So there are specific nutrients that are used throughout these stages, and we're going to just talk about a little bit more about what these stages consist on and what nutrients we require. Let's start with flowering. So flowering is basically when the white tip movement starts and you can start to see those bud swells. So I guess Emily has gone through the whole physiological stages in depth, but we're just going to talk about what are the critical outcomes at the end of the flowering. So by the end of the flowering, the total number of pieces of fruits have been set and total number of cells in those fruits have been set as well. So it's very important to optimize and understand the conditions for flowering for a healthy fruit development and maximizing those yields. And all those conditions are nutrient dependent, environmental condition, pest and disease dependent. So obviously us being a bit of a nutrient connoisseurs in the industry, it's important to understand how we can optimize the flowering outcomes by giving the right nutrients. Let's move on to calcium and its role in the flowering. So this slide, demonstrates the role of calcium early in pollen germination. Excuse me. So to achieve any outcome from flowering, we need to achieve optimal pollen germination. You'll see here, so once the pollen is released from the anther, it lands on the stigma, and the stigma then starts to swell. An enzymatic process begins to break down the wall of that stigma for that pollen. So the stigma will provide a supplementation of calcium to that pollen. So having, and obviously calcium is the main agent in that, but there's a bit of boron involved in this process as well. So having that availability of calcium and boron is very, very crucial for that pollen germination process. Once that, I guess the bit of the fertilization has happened, the pollen tube elongation is the next step, and calcium is involved in the pollen tube elongation as well. So that this is the key stage where we need to have adequate amounts of calcium that's available for the plant uptake as well. Other key traces or key elements required during that flowering, pollination, and fruit set phases are listed up here. So we're just going to skim through a little bit of the functionality of these nutrients. So manganese is a key one to regulate the potassium. It has been known to antagonize the calcium into the plants. So when we need that calcium availability and the functionality of calcium a little bit more, it's important to regulate the potassium with manganese into the plant. Next one's zinc. So obviously that's involved in cell wall membrane. That's one of the main functions of zinc, and it maintains the protein production. So it's very important to have zinc in the combination with calcium during that key stage. Then we've got boron, which is important for calcium uptake in the plants, as we all have heard many times. And it's also important for flower strength, elongation of pollen tube as well, and pollen germination as well. Phosphorus is an integral part of the ATP in the plant, and it's involved in the energy transports, the fuel that plant runs on. So phosphorus will also form the protein that will convert the other nutrients into the usable forms as well. So that's another key one. Moving to cobalt. Cobalt is important in root tip growth, and root tip growth in return induces cytokinin response into the plant. So obviously when going through that reproductive stages, cytokinin dominance occurs into the plant. So it's very important to have adequate amount of copper into the roots, and in the foliar as well. Copper is very important for pollination process, as it is required for pollen tube formation and cell wall elasticity. Then we've got seaweed, which is not necessarily a nutrient itself, but it's a source of available plant nutrients and source of secondary metabolite that I guess helps with triggering a bit of metabolites into the plant itself. So we've got calcium, and we've got all these nutrients that are key for that flowering and cell expansion, or sorry, cell division stage of the plant. Once the fruit or the fertilization occurs, the next step is cell division. So this process is where every cell in the final piece of fruit is created. So this is obviously Emily's slide from last week. I thought it was self-explanatory, and it was really good just to have a look at a bit of the process. So cell division transforms the flower over into mature fruit by rapidly multiplying the cells. It's very crucial for cell development and size. Timing, it's very, very important for us to reiterate that at the end of the 10 or 14-day window, the cell division completely stops. So it's important to understand that that 10 to 14-day window is very crucial from a nutrient availability point of view as well, having enough nutrient for the plant to help with that division, and which is calcium in particular, but we're going to just go over that again. Other factors that influence that cell division: climate, water availability, nutrients will affect the number of the cells in the fruit, and it will optimize the fruit quality. It will maximize the potential of the genetic capabilities of the fruit as well. Calcium. So this slide shows the function of calcium as a structural integrity in the cells. So it's a key component in formation of the cell walls. It helps cross-link the pectic molecules in the layers between the plant cells. So this cross-linking will provide rigidity, stability of the cell wall. So that's the key function of the calcium outside of that pollen germination and pollen tube elongation as well. It will also act as a secondary messenger in signaling events in environmental cues. I thought that was a very important point as well. So obviously, we've talked about calcium's role in cell division. So it's very important to have availability of calcium during that 14-day period, or however long that period is, fruit dependent, variety dependent. And it's important to ensure we have available form of calcium during that cell division period. This is an example that came out of University of Maine, where they identified a Honeycrisp apple variety, which suffered a bit of a bitter pit symptoms due to lack of calcium deficiency during the flowering and cell division stage. And that can obviously be monitored or regulated by applying regular calcium in available forms into the trees. Let's talk about application of these nutrients, calcium and other nutrients, via soil, which is a standard practice all across ag. So calcium, as we know, that it moves via xylem, and it tends to follow the transpiration stream into the plant where it moves with the water. So soil applied calcium generally moves and favors the leaves where the moisture is lost rather than the fruit. That's how it will be taken up. So uptake of the calcium from the soil will be dependent on the pH of the soil. So that's a bit of the optimal pH for the uptake. Soil moisture, so adequate soil moisture because it moves with the water through the xylem. So moisture is a main part for the availability and uptake of calcium. Transpiration rate as well, so that's temperature dependent, however much the plant is transpiring will impact on the uptake of calcium. Health of the root and structure, obviously, so that's a bit of a given with all kinds of nutrients. The health of the roots directly influences the uptake of nutrients from soil. Presence of other ions, so if there are other ions into the soil, as we may have come across looking at a lot of soil tests in recent times, that potassium, magnesium, sodium, presence of all these cations will impact on the uptake of calcium. So other nutrients overall, so limitation, there are a bit of a limitations that come from application of the soil, and especially in the periods where we need available forms of certain nutrients for a set amount or a period of time. So obviously so from the soil, the pH is a big part of the availability. So this is a bit of a recap of the chart we looked at a few weeks ago. Obviously, if there are extreme pHs in the soil, it will impact on the availability of micronutrients, calcium as well. So obviously, yeah, too acidic, too alkaline, no good for calcium availability. Mobility in the plants of certain nutrients will behave or move in different types of mobility. So especially calcium and the trace elements that we are looking at for the flowering period, they are either immobile or partially mobile. So it's hard to move them once they're deposited. So that rapid movement obviously is a bit of a limiting factor. Environmental constraints. So we talked about the root health, temperature. So if it's really cold during the early season in the soil, some of the nutrients need a bit of a ambient temperature to be, I guess, soluble and available for the plant roots. Moisture, if it's waterlogged, too dry, that can impact on that uptake of those elements as well. Competing nutrients, we touched on that as well. So I guess, yeah, that's a bit of a limitations when we are looking to apply the nutrients just from the soil. That's where feeding through the foliage comes into the play. So benefit of foliar feeding is that we can target the nutrients directly onto the foliage and the canopy. And by doing that, we can bypass the soil and interactions of those nutrients with other, I guess, factors. So all these interactions like nutrient antagonism, leaching, volatilization, fertilizations, the efficiency of fertilizers, we can bypass all those inefficiencies through soil. And we can deliver small and targeted amount of nutrients into the foliage. So obviously this slide comes out of John Kemp's webinar that he did for us in Wangaratta, and that's what I've kind of used because the information that he's delivered is amazing. So I guess that's how we can use or leverage the position where we can foliar feed certain nutrients in certain stages of the plant health or plant physiology. Pathways that the foliars are uptaken through the leaf. So traditional view is that, obviously John Kemp said that traditionally the nutrients are uptaken by cuticle or stomata of the leaf. But obviously more research shows that trichomes, vein, or even epidermal structures of the cell can uptake directly those nutrients. So obviously that there are pathways, there are multiple pathways from where the plant can uptake these nutrients. Variables for those plant responses. So all of these four categories, they will, I guess we need to consider all these categories or factors when we are applying through foliar. So first is formulation. So that's just how the the tank mixes in general, or the type of the nutrient application, the timing, environmental condition, the type of the crop, and yeah, a bit of environment, sorry. Formulation, so water quality. So water quality, when we're applying foliar, is very important. Generally watch for bicarbonates, because bicarbonates can make, I guess, the solubility of some of the nutrients, impact on the solubility of the nutrients. Usually it's recommended to use rainwater, or if the water is too salty, it's recommended to dilute the water with a bit of a rainwater. Water pH. So when foliar spraying, obviously, generally it is recommended to spray in with an acidic pH 5 to 5.5. Recommended to not go above 6. So that's a big factor when we are foliar spraying. Solubility. So leaf surface is a negatively charged area, and generally we need charged neutral molecule to pass through that leaf surface. So so that's a bit of an important point to see when we are looking into products to foliar spray, and we'll talk about it a bit later on into the presentation, that how the charged molecule, neutral molecule, will travel better or translocate better into the leaf. Chelated sources, so those charged neutral molecule, I guess, chelation comes into the play, and it neutralizes the cations and anions of the nutrients into the tank mix. So chelated sources can be carbon, molasses, amino acids, fulvic, humics, all of those sources that we are well aware of. Surface tension, spreading capacity. So I guess how much concentrated the spray is, and how well it spreads onto the leaf. So, and obviously when we are spraying pesticides, we need the nozzles to be quite tiny to cover the leaf surface entirely. But with nutrients, we have a bit more lenience with the droplet sizes compared to the pesticides. Let's talk about environment. So because the nutrients or the water is uptaken through the stomata, so it's recommended to look into the time of the day and the humidity while spraying any of the foliar applications. So generally, the 70% or higher is recommended for the humidity when we're foliar spraying. And that's when the stomatas are fully open and they can uptake the nutrients quite rapidly and quite quickly. Generally the time, early mornings, late evenings, obviously that's all dependent, but something to keep in mind. Higher dilution rate increase the microhumidity. So if we are going out and spraying during the day when the humidity is not just quite there, so just a high dilution rate will I guess make a bit of a microhumidity around the leaf as well. So that will help as well. Temperature. So as we all know, temperature is a very important part of the foliar spraying. So just make sure that it doesn't exceed 28 degrees Celsius, but obviously keep as low as we can. Wind is a factor as well. So 2 to 9 mph is generally recommended. Electrical conductivity. So the nutrients, how the leaf takes nutrients is a passive process as compared to active process through the roots. So it will just move from a high concentration gradient to a low concentration gradient. So it's a very simple process. So having the right EC in the foliar sprays is very important for the leaf to uptake the nutrients, or those sprays to move quite quickly from that concentration gradient point of view as well. Let's look into carbon-based inputs. So as we were talking about chelation, so most of the nutrients, as we know, are either cations or anions. So having chelation with carbon or molasses or any organic source will neutralize that charge that is present in those nutrients. And by doing that, it will increase the absorption of those nutrients, because it will be highly mobile and it will be translocated faster into the leaf. It will also have lesser phytotoxicity risk, because chelated nutrients have lower salt index. Chelated carbon will also save the energy from the plant because it obviously is bringing that organic carbon, and it will save the photosynthetic carbon expenditure from the plant as well. So it's just from that expenditure point of view, it is great as well. So just whenever we're looking into supplying nutrients by foliar means, it's very important to consider the nutrients or the products that come with that chelation. So these are the two main products that we in HybridAct recommend to our growers. So PowerCal is a sugar chelated calcium product. It can deliver calcium via phloem transport because it's a cal acetate form of calcium. It will deliver calcium to the growing form's location, and it will transport calcium from the leaves into the fruit as well. So obviously it brings the bit of that sugar chelation we talked about, and it also transport the calcium via phloem transport into the plant as well. OptiTrace Bud Max, so that's a chelated flowering mix that we generally recommend. Boron, manganese, zinc, cobalt, and moly. That's all the nutrients that we talked about are essential in that pollination flowering phase. So it's also a sugar chelation product, and it will maximize the pollen viability. So it's just good to, I guess, know that we've got those products that tend to work, I guess, based on the scientific facts as well. These are some of the research that has been done that I could find on the foliar application. So this one in particular looks at calcium and boron, foliar applied in Indian gooseberry. Obviously, the forms of calcium are not going to be the same as we generally recommend in PowerCal, but it's just good to know that there are some research out there that looks into the foliar application. So this one's calcarbonate with boron. It reduced the incidence of fruit deformation, blossom and fruit drop as well. So it lowered the blossom and fruit drop to a significant percentage, and fruit yield. So it maximized the yield of the tree with application of this particular rates. So that was good to look at. There's one study in blueberries. It talks about cal chloride. I know it's not the go-to recommendation for calcium, but it's just good to know there are studies on cal chloride as well. It increased fruit firmness up here and better antioxidant response. Not saying that obviously there might be other better studies in blueberries that's been done other than cal chloride, I guess. This is done on cherry trees, early calcium load. So this again is done with cal chloride on dormant buds on quality of sweet cherry fruits at harvest. So this is the response flower buds. So what they said was quite interesting. The higher calcium content was detected in phloem and flower buds, and not in vegetative buds through that application of cal chloride. Then we've got the micronutrients. So that was just done in lychee in India, combination of cal nitrate, boric acid, and sorbitol. So it affected the initial fruit set, number of fruit, and yield and fruit weight as well. Foliar boron in olives increased perfect flower and fruit set. It did not impact on pollen germination, but obviously that impacted on the fruit set, all dependent on what the quantities and the forms of nutrients are. Let's dive into some of the common concerns that I've come across just by talking to the several teammates that we have. So phytotoxicity risk is something that we come across from the growers. Chelated products, as we talked about, they have low salt index. It comes with carbon. They are highly mobile and more stable. So once you apply them compared to non-chelated nutrient products, they are less likely to build on leaf surface. And they have high movement, high translocation rates, and obviously low ionic strength, and which will contribute to lesser salt load as well. And it's very important to know what is in the in the water tank as well. So if we are, I guess, mixing a bit of pesticides or fungicides with those nutrients, it's important to know what the forms are and how they can impact on that phytotoxicity as well. So it's important to just use chelated products when spraying with nutrients, diseases. So obviously that's one that comes out of concerns about high humidity or temperature while spraying during flowering. So it's important to monitor the weather conditions, avoid spraying when it's extremely humid conditions, watch out for those, I guess, slightly better weather conditions, and have a good IPM strategy in place. This then came out from Smithy, impact on the bees. So obviously, as we talked about, avoiding harmful chemicals into the tank mix that will impact on the bees, and spray whenever the bees are less active. So that's early morning or late evenings. And I probably should look into more of chelated products and nutrients on impacts of the bees. So I think, yeah, that was just a few things that came out. So take home points that we have: calcium is very crucial for pollen germination, pollen tube formation, and cell wall formation. There are these trace elements that we talked about. They will play various essential roles during bloom and pollination stages. It's very, very crucial to have this nutrient present during flowering in available forms, and not all of those forms are quite available from soil applications, or they can't be uptaken within that time frame if you have extreme pHs or moisture or temperature, other factors that come into the play. So we can look into foliar applications to overcome some of those limitations from soil applications. So it's very, very important to look into spraying chelated forms of nutrient under right conditions to have high efficiencies of these spray. So to answer the question, should we spray during flowering? Hell yeah, let's do it. Any questions?
Yeah, well done, KJ. Plenty of information there. So it's interesting when we talk about calcium and how hard it is to actually apply it right or use it right, because there's so many limitations, I guess, at that time of the year from a soil point of view, whether it be other anions, pH, or the fact that we don't really have a tree cycling because it's not drawing so much from the soil. So I guess that's where it gets back to the post-harvest story, and senescence process is super important, as we know, because all this activity based around our flower and bud development is really driven through storage nutrients or nutrients stored. But it does show, particularly in some of those studies that you brought up, that some of those earlier applications of those various nutrients, being calcium and trace elements, do have an impact. But it's also interesting to see that when you actually put them with a carbohydrate, how it gives it that buffering effect or chelation to actually make it way more useful or plant useful when it needs to be. And then we've got the bicarbonates. It could be in the water tank from the water source, which can have a limiting factor, not only with nutrient, but the actual ag chemical as well. We've got the humidity. We've got things like EC. We've got the temperature from, once we get over 28 degrees, and that's when we actually do have a leaf on a plant. We know once it's over 28 degrees, it's not actually going to be photosynthesizing anyway. It goes into that photorespiration phase. And the other thing is, by adding that carbohydrate to those types of nutrients, it saves plant energy as part of that conversion, a bit like that nitrogen story with urea and being an amine form of N. So it's when we sit back and say bitter pit is a calcium disorder, well, yes, it can be a calcium disorder, but have we, as a grower or an agronomist, nutrient agronomist, given the calcium the best opportunity to actually get into the plant? Because there's so many of those factors that can be limiting, and it's one of those key nutrients that we really do need through that flower bud development stage. So it's super interesting discussion. I think you really covered it well. So well done. That's all the observational questions that I've got. Has anyone else got some questions for KJ?
Thanks, KJ. I just wanted to, just got in front of you, Woody, sorry. Sorry if I missed it, KJ, but I'm just curious if you came across any research on absorption of nutrients through the actual petal. Because I don't know if you remember when we were in the post-season review, I just stumbled upon that comment from John Kempf somewhere when I was Googling when we were, maybe it was when we were having the Joel Williams session or something. I haven't followed it up since, so, I was just curious if you came across anything. And yeah, because I guess developing this program last week and having a chat to Linton, he was like, oh, I usually try and keep out of the right in the flowering window in the vineyards. So I sort of shifted some of my recommendations just outside that window. But yeah, just curious if you came across anything or if it is still a danger to, yeah, anyway.
I think that's a really good point, Nate. That was something that I was thinking myself. I haven't come across anything, or I haven't really looked into the absorption through pedal, but I think that's a really good point. And I've come across those concerns as well in the vineyard that they just don't want to, I guess, spray during the flower because it causes that rossets formation. So yeah, I'll just definitely, good point. I'll look into it, see if I can find any information on that pedal absorption as well.
Yeah, cool. Likewise. Yeah, I'll suss it out. Thank you.
Am I misunderstanding the thing with other nutrients versus calcium? Well, I thought the maximum temperature we should be spraying at is 25 degrees rather than 28.
So this one, Woody, is just, it came out of Joel Williams' webinar, and it was very generalized. So 28, he said, just overall with all the nutrients. So considered, for example, it just wasn't specifically for calcium, but he did say go as low as you can. So 25, 22. I'm not 100% on what's the absolute maximum that you don't want to surpass. Is it 25 or is it 28? 28 does sound on a bit of the edge of the
I heard many years ago when I worked for Advanced Nutrients that they had someone spraying cotton late into the morning. And they reckon that when they went back and did leaf tests later on, they found no response when they went over 25 degrees from boron applications. That's what I get back to, but yeah.
Yeah, interesting.
One might be able to tell me otherwise.
I'm sure they'll be nutrient dependent.
I think the other thing which happens after that is it's like, it's okay, we can spray before 28 or whatever, but it's like, what happens if it gets to 35 or 6 as part of that day? And I think that's where if the temperature is elevating significantly, you might still be under 28 or around 25 or whatever, but it just gets back to what does the rest of the day bring? Because if you're going to spray, you really need to have the plant actually do some of that work before it actually turns off through the day on a foliar application. So I guess the rule of thumb is if it's going to be a stinking hot day, you need to be thinking about, do I want to spray even if it is at, say, the right temperature window, because it might only be there for a minimal time of the day, but the rest of the day is going to be stinking hot. So how valuable is that sort of a day to you from a spray application point of view?
Yep, and more relevant in Queensland than elsewhere. Yeah,
so it all gets back to that humidity and daytime temperature after it as well, not only before, based around the length of period of time. But yeah, good observation though, and obviously, yeah, lower temperature is better. So if you've got that opportunity, certainly try and structure your spray applications around the right temperature window, particularly based around the daytime length as well. Slower drying time is always going to be better. But I think the other thing you touched on, Nate, about the flowers, and I think the missing link in some of this information that is out there as the urban myth is in a lot of cases in the past, we've had non-chelated products which have been sprayed on flowers and it's created issues. And there's been other things putting tanks, and there's water as well. It sits in the background of some of that story. And when we come along with products which are actually chelated or have carbohydrates with it, we see a different result because we have that buffering or that salt index reduction versus something which is more of a sulfate type thing, which can, or a chloride, which can burn through that flower period. So I think that's something else to consider. And I know it's a bit anecdotal, and we probably need some reference to back that up, but that's probably what we've seen based around what we've done in the past with the hybrid ag products into our bud and flower program, because effectively it's sat in stone fruit and pome fruit for a number of years without any detriment. But bearing in mind, they are not non-chelated products which we're using. So it's a very different place to what other standard nutrient chem will be, is the position that we've taken.
That's
for obvious reasons, which KJ have specified through this discussion today.
Yeah. Is there any potential to disrupt pollination, do you think?
I don't think so. When I say I don't think so, I think it gets back to whether you're stopping bees from flying or hurting bees. And I don't think that nutritive input, which we're using, and you do hear some people say that boron and bees it's not a good mix. But once again, I think it's a different type of mix of boron which they're using versus what we've got to offer.
If it's just loading up on the surface, and interesting. Thank you.
Yeah, I see you're there, Ham. You got any comment on some of that stuff we just touched on now? Maybe not.
Sorry, just trying to find my mute button.
Yeah.
The probably two main comments. I know John Kemp talks about leaf temperature of 23 degrees. And again, I think what you referenced earlier about the temperature of the day and what's happening for the rest of the day is very, very relevant. And also even the humidity and things like that, because they make such a big difference. We look at the temperature as one fairly isolated factor without taking into account all the other things that might be happening on that particular day as well. With the spraying of calcium in flower or nutritive things full stop, I think the other thing to bear in mind is they've all got a food source, normally in the form of a sugar, which if anything could well be attractive to bees anyway. And certainly from what we've seen over the years, that it doesn't scare them away by any stretch of the imagination. And as we know, without calcium, or the lower the calcium content in the actual pollen tube itself, the lower the opportunity is for that flower to produce the best flower to pollinate, or the most pollinatable outcome. So they're probably the only thoughts that I had. Other than that, just a really, really good presentation as always.
Yep.
Another thing that's relevant is light intensity. So I know that you come the end of September up here, and it gets pretty hot then, October, and I'll actually put up heavy shade cloth over my veggie garden, and the plants don't stress as much because you've still got the heat. So you've still got the heat and the humidity, but you don't have the light intensity on the leaf surface, so it reduces stress.
Yeah, so you're creating a different microclimate by doing that effectively.
Yeah.
And that's what we see. That's what we see under netting and particular rain covers, that we do create a different microclimate, which has an effect or an outcome to that fruit which is growing underneath it. And sometimes it can be negatively, depending on the season. Or I shouldn't say negative, it can create a different outcome, and that's where you just got to be across what is going on from a plant function point of view with that different microclimate.
I'd also realise plants develop their chlorophyll and everything, whatever the conditions they develop in, if that changes, it actually affects the plant too, because so if you have high light intensity and then drop to low intensity, yeah, the plant is actually growing depending on the amount of light when it's being formed.
Well, absolutely. The photo period of the day has a reference or an outcome with the photosynthesis or photosynthetic capacity of the plant. If that's been reduced, obviously we reduce that opportunity to capture and create and draw from our roots. Effectively, we have a bit of a brown out. It's like a grey day earlier in the season. It's a similar type thing. Reduce that capacity of that light penetration will change that photosynthetic outcome for sure.
Yep.
All right. Any other questions? Em, you got any questions at all?
No questions from me, thank you.
Okay. Righto. Well done, KJ. That wraps that one up. Thanks for so much.